Hello, I'm Dr. Aizaz from MedicoVisual.com, and in today's visual lecture we will talk about the basic concepts of development of Rhombencephalon. So let's start from the very basic concept that here we have a brain, and this is AI generated picture of the brain, of course not anatomically correct, but here I want to show you something. So if you look carefully, this brain, it actually resembles a tree, and here you can see that tree has mainly two parts, the tree stem and the crown. So this one is the stem of the tree, and other part other than the stem, all other parts of the tree, they are the crown. They consist of branches and leaves, so they are the crown of the tree. Similar to that, our brain can also be divided into two parts. Here we have the brain stem, so similar to tree stem, we have the brain stem here, and we do not have crown of our brain, but instead we have the other part, which is termed as collectively as higher centers. Now higher centers consist of the cerebrum, the cerebellum, and some diencephalon derived structures that we will discuss later. And brain stem consists of midbrain, pons, and medulla. Let me explain these things to you a little bit more clearly. So for example, if I draw a simplified Dr. Najeeb style diagram of a brain, so here we have, this one is the midbrain, and then we have, okay, let me draw it for you. So this is the midbrain. Let's use another color. This one is the midbrain. Okay, then here we have pons, and then here we have medulla, and of course this one is the spinal cord, and these are the higher centers. And please don't forget that higher centers also include cerebellum. Now here the interesting thing is that all these three structures, which include midbrain, pons, and medulla, these three structures, they constitute the brainstem. While cerebellum, it is not the part of brainstem, it is the part of higher center. It's not the part of brainstem. Please keep in mind, cerebellum is not brainstem. It's not brainstem. It's the part of higher center. But the cerebellum is often discussed with the rhombencephalon, with the brainstem actually, because it is derived from the rhombencephalon, right? Let's write it here. Rhombencephalon. Rhombencephalon is divided into two parts. The metencephalon, we have discussed these things previously as well, metencephalon and myelencephalon. Now the metencephalon, it forms the pons as well as cerebellum. So pons as well as cerebellum, cerebellum and pons, they are derived from metencephalon, while the medulla oblongata, it is derived from myelencephalon. And midbrain is derived from mesencephalon, which is a separate brain swelling that we will discuss in detail later, that how the brainstem develops. But keep in mind that the cerebellum, it is also, although it's a higher center, but it is also derived from the rhombencephalon, specifically from the metencephalon part of the rhombencephalon. Now these things are very important for MCQs as well as in the short essay questions and other questions, and for the concepts as well, that what structure forms what structure, right? What layer forms what, and so on, what swelling forms what structure. These are often asked in embryology. I think this is the crux of embryology. Our aim of learning everything is ultimately to master that what structures are derived from which layer or what structure, what embryonic structure, and so on. Now you don't need to cram any of these things. we will see one by one that how these structures develop. First, we will see how exactly the Rhombencephalon develops into forms the Pontine flexure and how it is divided into meten and myelencephalon. We will see in quite detail and we will try to understand visually. Then we will see that how pons and medulla develop, and we will also learn cerebellum. I think we should first discuss the medulla and pons and midbrain, I mean the brainstem as a whole, then we will come to cerebellum. But don't worry, stick with me and I will explain each and everything in a great detail to you, and hopefully you will not get confused. In one of our previous video, I had shown you the formation of Pontine flexure and I've told you that it's not a correct animation. Now we will watch the correct animation of Pontine flexure. That won't be that much simplified, but you will be able to understand it at this point now. So here is the neural tube, and you can see it is somewhat like this, shaped like this, rhomboid shaped, and you can see that it is divided into certain parts. So let me show you exactly that. Here we have the primitive spinal cord. It is basically the primordium of spinal cord. Then here we have the rhombencephalon, this bluish one. This is the rhombencephalon, and we will see why it is called rhombencephalon, that we will also see today. And then this brownish one is a part of mesencephalon, and here actually I have removed the other parts, and here we have the diencephalon, and then we will have telencephalon here. So diencephalon and telencephalon are basically the part of prosencephalon that we will see later, and actually I have removed it in this 3D model to keep the things simpler. Now what actually happens now to understand how the Pontine flexure forms, here you can see that this is basically the roof of, here this one is the roof of Rhombencephalon. Now what actually happens that this roof splay opens, it widens up, right? It widens up, it spreads, and they say in textbooks that it opens like a textbook. So let's see this beautiful animation to understand what is going on. So here we have this roof. In the middle part of Rhombencephalon, it opens like a book, like this. It's a play opens. So you see what is going on here. So now this huge, relatively larger cavity is formed. Now this cavity is basically the fourth ventricle, and here this roof plate is now actually the roof of the fourth ventricle. Now the lumen of neural tube in the spinal cord part, it is relatively much narrower. And here also this lumen is also much narrower in the mesencephalon region, but here the lumen is relatively larger. So this is basically fourth ventricle, and this is, which is still narrow, it is the central canal of spinal cord, and we have this part is also narrow inside the mesencephalon. This lumen, it is called aqueduct of Sylvius. Aqueduct of Sylvius. Now I have removed a few things in this 3D model previously to keep things simpler. Let's add those things. So if I add all those things, now you can see here, you can actually see the cross section of developing spinal cord, that the neural tube, it forms 3 layers. Let me explain these things to you again, that here there will be the epithelial layer, which is called ependymal layer, or it is also called ventricular zone or ependymal zone. So epithelial cells are here forming the ependymal zone. Then outside it, here there will be this main zone which consists of the neurons. So this is mental zone, and outside it will be the nerve fibers. Now the mental zone, which consists of the precursors of neurons or developing neurons, it is rearranged or reorganized into two aggregates, two columns, or two types of, actually four bilaterally they are. So actually four columns are aggregates of developing neurons. These are called alar plate. Dorsally we have alar plate or alar plate, and basally or ventrally we have basal plate. And by the way, this structure as a whole, this structure, which consists of, let me again explain it to you, it consists of mainly the ependymal zone, and here we have the pia mater. Of course outside it is the pia mater. This greenish one is the pia mater. So pia mater, along with that we have this part of, this top part of or basically dorsal part, not exactly top, we should say the dorsal part of ependymal layer, ependyma, and some nerve fibers are there as well, some white matter is there. So this white matter layer is the marginal zone. So mainly it consists of ependymal zone as well as the pia mater, and there is little bit of nerve fibers in the marginal zone as well. So this is basically the roof plate. And this configuration, it continues throughout, it spans throughout the neural tube, this developing neural tube. So it's like that all around. You can see here as well in the mesencephalon, initially in the rhombencephalon as well. Initially, later on things will change, but initially it's like that. So now again, if the roof plate, here is the roof plate, if it again supply opens or widens up, then what will happen? You see what is going on here. So you see the roof plate, it widened up a lot, and the location of these columns, the alar and basal plate, it also changed. Just focus on these plates now. That basal plate, it remains almost at the same place, medially lying. But what about the alar plate? That alar plate, it is initially dorsally lying, but it comes to lie slightly ventrally and lateral side, towards the lateral to the basal plate, like this. So this configuration becomes apparent in this region of the rhombencephalon. It will become more clear when I show you a cross section. So here we have where is that cross section? So here we have two cross sections. Here we have the cross section, and the other one is also similar. I will show you what is that. So here you can focus again that let me show you the structure. These are the alar plate, and these are the basal plates. I don't think I need to label it again here, but the important thing is that this one is the roof plate. Now what is going on here? It's worth repeating that this roof plate, it widens up like this, it splays open, it spreads laterally, and as it spreads laterally, these alar plate, they also move ventrolaterally, mainly laterally, but slightly they shift ventrally as well. So let's see the animation again. That here is that full 3D model, and here is just the cross section. So let's watch it again. Okay, let me show you again. Now just focus on this now, that the focus on roof plate mainly and alar plate. There is not much changes in the basal plate, but roof plate, as it widens up, the alar plate, it moves ventrolaterally. So now basal plate is lying medially and ventral plate is lying laterally. And here I want to show you another thing as well. So initially you see that at the lateral wall, in the lateral wall of the neural tube, this developing neural tube, here you can see that we have the lateral the groove in the lateral wall. You see shallow grooves in the lateral wall, and these grooves are basically the sulcus limitans. Sulcus limitans demarcate that here there is the ending of alar plate, and here the basal plate starts. So it demarcates the alar plate from the basal plate. Now the position of sulcus limitans also changes. You see the animation again, that here again the position has changed, but still the function of sulcus limitans remain the same. That the goal of sulcus limitans is to separate the basal plate from the alar plate, or alar plate from the basal plate. And by the way, this one is the medial sulcus. In the midline we have this not medial, it's called median sulcus, and this one is also, and this one is also, both of these are sulcus limitans. And now this was the situation, this is now the situation of roof plate. Initially it was very narrow somewhat here, but it has widened up, it has spread laterally, and this is the situation right now. This is very important to understand. That is why I'm stressing so much on this. If you can understand this, then you can understand whole of the neuroanatomy of medulla and pons, and even to some extent midbrain as well. So this is the most important thing to understand. Okay, now let me show you another thing as well, that with the formation of this pontine flexure, this is basically the pontine flexure. Well, actually in textbooks they usually remove this roof of fourth ventricle so that things become more clear to you, and I'm gonna do the same as well. So here I have removed the roof of fourth ventricle, and now you can see inside. This is the diagram that you are used to looking at the textbooks. And by the way, this one is the sulcus limitans, this is the median sulcus, here also sulcus limitans and median sulcus. You should be able to appreciate this clearly on this 3D model as well as in the cross section. By the cross section, of course, I mean that I have cut it somewhat here, a section from here like this, and we are looking only at this section. I hope you know what we mean by the cross section, but still some people might not be knowing, so here I have explained that as well. So anyhow, now what this one is actually the pontine flexure. Let me exactly tell you that this is the flexure, that it's like this. This is the situation, this is the flexure, this one specifically. Here is actually this at the pontine flexure part, this one is widest, the fourth ventricle is widest. And by the way, if you look carefully, this fourth ventricle and this rhombencephalon, this opened cavity, it is somewhat looking like a diamond. Yeah, so it is looking like a diamond or rhombus shaped. So that is why this part of the brain, it is called rhombencephalon. This part of developing brain or embryonic brain, it is called rhombencephalon. Rhomb for rhombus, so because it's rhombus shaped, so that is why it is called rhombencephalon. Encephalon means brain, so it is rhombus shaped brain, so that is why it is rhombencephalon. With the formation of rhombencephalon, the brain has now been divided, not brain, I mean the rhombencephalon has now been divided into two parts. One part, this exactly is the point of pontine flexure. This is the point of pontine flexure. Some people also like to call it rhombic flexure, but mainly the common name is pontine flexure. So rhombic flexure or pontine flexure. So above this flexure, I mean cranial to this flexure, is this part of rhombencephalon is metencephalon, and caudal to it, this part of rhombencephalon is myelencephalon. Myelencephalon will form medulla oblongata, and metencephalon will form the pons as well as cerebellum too. So now let's move forward and let's try to understand what happens next. So here are the two cross sections. this upper one is the section of metencephalon. This is the section of metencephalon, and this lower one is the section of myelencephalon, which will form medulla oblongata. So what I mean by this is that okay, let's suppose here we have the, let me show you. So we have this flexure, Pontine flexure here. So the upper section is of this area and the lower section is of somewhat this area. So this is metencephalon and this is myelencephalon section, right? So again the roof plate widens up, and then what happens that in case of metencephalon, not with myelencephalon, but in case of metencephalon, the alar plate, it grows dorsally at its lateral end. So somewhat here it grows dorsally, this part it will grow dorsally, and this growth will then thicken up to form a lip-like structure, and here as well. Now let's see the animation to understand this. So like this, it has formed this lip-like structure. So what is going on here? What is this lip is, by the way, called rhombic lip. So hold this lip here, this one and this one as well. These are the rhombic lips. These are bilateral structures, and of course please remember that the rhombic lip, they are made up of the thickened dorsolateral part of the alar plate. So for this is dorsal side, of course this is dorsal side, this is ventral side, and this midpoint is of course medial, and this is lateral. This is lateral here also. This side is lateral, ventral, and then dorsal. So here is the lateral and here is dorsal. So this end, this is the dorsolateral end, and this is also the dorsolateral end. So in textbooks they say that the rhombic lips, they are formed by a thickened dorsolateral part of alar plate of the metencephalon part of rhombencephalon. So now you really understood this statement. And also please note that the rhombic lips, they are not just formed by the thickened dorsolateral parts of the alar plates, but they also include this part of ependymal layer as well as this part of the pia mater which is surrounding it. So this structure here as well. This structure is the rhombic lip, and rhombic lip is actually the precursor of cerebellum. Later we will see how exactly the cerebellum develops, but today we will not go into development of cerebellum, but basic concept you should be very clear at this point that this rhombic lip, it is the precursor of cerebellum, while the other part, which includes this ventromedial part of alar plate as well as the basal plates, the remaining part, it forms the pons, while this part, which is rhombic lip, which is made up of dorsolateral part of alar plate and pia mater and ependymal layer, this rhombic lip is the precursor of cerebellum. It's very important to understand. And if I zoom it out a bit and I show you the animation again, so here you can see that this thing happened with the metencephalon part, but not with the myelencephalon. Now what happens that these columns are then split into nuclei. Let me explain these things. So let me draw the column. For example, here we have, let's just draw the okay, let's assume that this is the basal plate. So if I draw this column of basal plate like this, now this is just about the brain stem. In other parts, in spinal cord, this is not the situation. Of course, in the other part of the brain also, the situation is a bit different. Now what happens that a lots of nerve fibers are there. There are lots of connections that are within the brain stem in the brain, and there is lots of white matter, lots of axons going on here and there forming different connections. So that is a reason, that is a speculative reason that this column, it cannot, these columns, they cannot remain continuous. They become discontinuous. And they form groups or aggregates of neurons, actually neuronal cell bodies. So rather than being this continuous column, for example here it may form a small aggregate of neuronal cell bodies. Then here it may form another aggregate of neuronal cell bodies, and so on. It may form another aggregate, and in this way different aggregates of neuronal cell bodies are formed, and this continuous column, it becomes discontinuous. You see, in the development, during the development of spinal cord, we had seen that there was that continuous columns. There was the continuous column, for example the lateral gray column, the ventral gray column, and we had the dorsal gray column. But here we do not have columns but aggregates of neuronal cell bodies. Clumps are aggregates or collection of neuronal cell bodies which are called nuclei. They are called nuclei? Well, Dr. Aizaz, previously you said that the aggregates of neuronal cell bodies, they are called ganglia, and now you are saying they are called nuclei. What's wrong with you, Dr. Aizaz? I think you need to study again. You need to clear your own concept first before teaching us. I know some of you might bash me for this, but let me tell you, both of my statements are correct here. Here I am not wrong. Trust me. The groups or aggregates or cluster of neuronal cell bodies, they can be called nuclei, as well as they can also be called ganglia. Both of the statement are correct. But if such aggregates are in the central nervous system, then we call them nuclei. But if such aggregates are in the peripheral nervous system, for example, we had talked about the dorsal root ganglia. So in that case, such group of neuronal cell bodies, such aggregates of neuronal cell bodies in peripheral nervous system, they are termed as ganglia. In central nervous system, they are termed as nuclei. Of course, at the histological level, there are other differences as well. For example, in ganglia we have that satellite cells. In central nervous system the situation is a bit different. At histological level we have differences, but the basic thing which differentiates the nuclei from the ganglia is their location. Central nervous system nuclei, peripheral nervous system ganglia. Remember this. Both are aggregates of neuronal cell bodies. So please note that. And now again, these columns, alar plate as well as basal plate, and in both of the sections, they will be converted into several different collection of cell bodies called nuclei. And in case of metencephalon, we will have this rhombic lip, but we will not have the rhombic lip in the myelencephalon. Other things will remain quite similar. And we have about 7 of such groups of nuclei, and we will talk about that what are these types of nuclei in the upcoming part of this lecture. And in later lectures we will also name these. We will see the examples, and we will see that what cranial nerves they do supply, and other details as well. So now let's try to logically understand that what are these nuclei. And by the way, I must mention here that here is of course the sulcus limitans. So medial to sulcus limitans, all these 3 nuclei. Here also this is sulcus limitans. So medial to sulcus limitans, all these nuclei. And here as well, all these nuclei are derived from which plate? Basal or alar plate. They are derived from basal plate. Of course, basal plate is medial to sulcus limitans. So these are derived from the basal plate, and those that are dorsolateral, to this sulcus limitans here as well, these are derived from the alar plate. So this you must understand, this is derived from alar plate and this is derived from basal plate. And of course, please don't forget that we have this lip here which will form the cerebellum. It is called rhombic lip. Now, to understand those nuclei in the rhombencephalon region, myelencephalon as well as metencephalon, let's understand the type of neurons in the spinal cord. Well, Dr. Aizaz, I came here to learn rhombencephalon, not spinal cord. Oh, please bear with me. I know you must be frustrated by such a long lecture, but please keep in mind you will learn it forever. So let's understand from its core. Okay, so here we have, okay, let's draw it from this side only. That here, and this is the basal plate derived ventral horn of the spinal cord. It's a section of spinal cord. Now here from this group of nuclei, it's actually a whole column. It's not a ganglia or it's not a nuclei, it's a whole column, continuous column, which is actually the ventral gray column of the spinal cord. So ventral gray column or ventral gray horn of spinal cord, in that we have the neuronal cell bodies and some of their axon, actually all of their axons, they come out, and what will happen that they are going to supply, let's suppose, a skeletal muscle which is derived from the somites. We have discussed these three things previously in another lecture as well, actually in a lecture series that I will share in the description. So anyhow, here they are innervating this muscle, and the order from the CNS will be conveyed with the help of this nerve fiber, with the help of this axon towards the muscle. Let's suppose we want to move this muscle, so order will move from the central nervous system towards this effector organ. So this is basically an afferent neuron. It is basically your afferent or motor neuron. We have, of course, many of these neurons going to muscle and to other structures as well. So this is an afferent neuron, and it is going to muscle, skeletal muscle, which is derived from the somites. So that is why it is a somatic afferent. And it is a common type of neuron, nothing special about it. So it's a general somatic efferent. General somatic efferent type of neuron going here, and this gray horn, is also the general somatic efferent type of horn, right? Gray horn. Now if we come to this dorsal side, here we have this neuron. Actually there will be some dendrites, and actually there is the dorsal root ganglia, but let's just ignore to keep things simpler. So here we have, let's suppose, skin, a simplified depiction of skin. So now actually some sensory, from the sensory receptors of the skin, some neurons, their peripheral processes, they will move towards the spinal cord, and here actually they will form these are basically pseudo unipolar neuron. Here there will be such aggregates of their cell body forming the dorsal root ganglia. So anyhow, it is going towards the, what is this, towards the dorsal gray horn of the spinal cord, like this. It is going towards the dorsal gray horn of spinal cord, and let's use another color here. It will terminate somewhat here. It may either terminate here and then it will cross. There will be another neuron which will cross, or it just may go upward as a white matter tract called dorsal white column. Anyhow, we will not go into those details. Here what I want to highlight is that this is a afferent, not efferent. It's a afferent type of neuron because it is taking sensory information towards the central nervous system, not away. It is taking information towards the central nervous system. Such type of neurons are afferent neurons. Secondly, it is taking the information from superficial tissue, for example skin, or even from the muscles as well. So there may be another similar neuron which is taking information from the muscle as well, like this, and it will also become the part of this, for example proprioception information. So anyhow, this is afferent and it is taking information from the muscle. So it is somatic. It is somatic from the superficial tissue. It is somatic. It has nothing to do with deep viscera. It is somatic, and of course it's general. So it's a general somatic afferent. So we have general somatic afferent types of neurons here, right? And then we have two other types as well. So let's understand those as well. Okay, so let's draw a viscera here. So it's a part of GIT. Now what will happen that, let's suppose we have some glands in here. In this part of viscera we have some simplified depiction of glands here as well. Now let's suppose we want to stimulate these glands. I mean the nervous system wants to stimulate these glands, and it wants to increase the secretion of these glands, or it want to, let's suppose, act on the rate of peristalsis. So for this we have here, this is the lateral column. So this lateral column is basically in T1 to L3 region, right? So anyhow, from here some neurons, their cell bodies are here, but their axons, they will join this part like this. Ventral root, this is by the way called ventral root of spinal nerve. It will join the ventral root and ultimately will become the part of spinal nerve, and ultimately it will come here to supply these glands and this viscera as a whole, right? So now these are coming towards an effector organ, glands in this case. So these are efferent, right? So these are efferent. Secondly, they are supplying the viscera, so they are visceral efferent. They are visceral efferent, and of course there is nothing special about them, so they are general visceral efferent types of. So here we have general visceral efferent types of neurons, here we have general somatic efferent type of neuron, and here we have, this one is the general somatic afferent type of neuron. Finally, of course, we have some receptors within the mucosa or internal epithelium of this deep-seated viscera somewhat here, right? So from these, the stretch and internal situation, internal chemical milieu, and all these stimuli, they are sensed by yet another type of neuron, yet another type of sensory neuron here. Okay, let's draw it with this color. So these are sensory neurons, so they will join the dorsal root like this. They will join the dorsal root and ultimately they will come to lie here in this region. Right here they will synapse, or they may go straight upward. Anyhow, they will come to lie here in this region. This region is basically the ventral most part of dorsal horn. It is the ventral most part of dorsal gray horn, or we can also say it is the ventral most part of dorsal gray column of the spinal cord. Now, typically, when we talk about the lateral gray column, this is the lateral gray column. This one is the lateral gray column. Structurally speaking, it forms this typical lateral bulge in the spinal cord. So, lateral, if we talk about structurally and anatomically, if we talk about this lateral gray column, it consists of only neurons that are general visceral efferent. But functionally, both of these are supplying the viscera, and both of these are the part of autonomic nervous system. They are involved in autonomous regulation and unconscious subconscious regulation at subconscious level. Usually you are not aware of, body is doing it automatically. Body is automatically controlling the rate of secretion of glands and rate of peristalsis and all these things. These are automated functions going on in your body. You are typically not consciously aware unless something is wrong going on here. Of course, there is if visceral pain inside the viscera, there is inflammation, then of course there is conscious perception. But typically, of course, it's an automated system and it is a part of autonomic nervous system. So functionally speaking, both of these can be considered as a part of lateral gray column, but structurally because only this part is forming the bulge, so many authors, they only consider these group of neuron which are the general visceral efferent types of neuron. Only these are considered the part of the lateral gray column by most authors, but some authors also consider these general visceral efferent type of neurons as well. These one they are also considered as a part of lateral gray column by some authors. Now, if you go with the majority, then the lateral gray column of the spinal cord is only derived from the basal plate. But of course, if we consider this part as well, then it is derived from both. This part is derived from basal plate, alar plate. This part is derived from alar plate and this part is derived from basal plate. So if we consider both, then it is derived from both of these structures. And by the way, this one is now the spinal nerve. This is the spinal nerve. Whole this is spinal nerve. It consists of different types of nerve fibers. Whole this is spinal nerve, and it has two rootlets. So one is this root, which is the dorsal root. This is the dorsal root. Both of these are actually rootlets of spinal nerve. These are not rootlets of spinal cord. These are the rootlets of one nerve, this specific spinal nerve. This is like a stem, right? So it is having the different roots, so different components of the root. So this one is the other rootlet, which is motor rootlet, and it is named as ventral rootlet or ventral root. So this is the ventral root, this is the dorsal root, and they combine to form what we call as a single spinal nerve. Of course, at different spinal levels at each spinal level we have a single spinal nerve. Of course, it will then divide into ventral and dorsal rami. We will discuss those details not in this lecture. We have also discussed previously in another lecture series on general visceral efferent, somatic efferent, and all these things. Now you understood all these structures. Let me write it again quickly, that this is the general somatic efferent, this is general visceral efferent, this is general which one is this? This is general visceral efferent, and this is general somatic efferent, right? So these are the neurons that are there in the spinal cord. Now you just assume, of course it's not correct, it's an incorrect animation, but it will help you understand the concept. Now you assume that the spinal cord supply opens, its roof plate supplies open. So here is the roof plate. If this roof plate supplies open, then what will happen, that all these neurons, all this group of neurons, their location, their configuration will change. Let's see the animation. Of course, it's not correct. Technically, it's an incorrect animation. Don't tell anyone that Dr. Aizaz says that Rhombencephalon is derived from spinal cord. No, it's not derived from the spinal cord. It is just for the sake of understanding that you see that these alar plate derived neurons, neuron collection of neurons, they came to lie ventrolaterally like this. So now you can again understand, and by the way, these basal plate, they remain medially. Between them is the sulcus limitans. And now you can again label these very easily. So here we have this general somatic efferent. Here we have general visceral efferent, right? Then we have general visceral afferent. And here we have general somatic afferent. These are derived from, this is the sulcus limitans. This one basically, this is the sulcus limitans, and these are derived from basal plate, and these are derived from alar plate. Now actually it's not like spinal cord. Of course, rhombencephalon and brainstem in general, even our whole brain, it is special, and it has some special neurons as well that we will talk about in a bit more detail later. That, and by the way, this confusing terminology, the general visceral afferent, blah blah, all these things, I was about to say all this shit because it is very confusing for medical students. So everything related with this classification, I have decrypted it in a bit more detail in a lecture series. Please watch that as well if you are still confused about this terminology. So anyhow, we have some more special nuclei here as well. So we have one of this here that develops again from the basal plate, and this is special type of neuron, right? It is special. And what is its function? Actually, it is supplying the muscles that are derived from pharyngeal arches. So for example, what is the muscle? Okay, muscles of facial expression. You remember that? So muscles of facial expression, of course, it is one of the example. Besides that, we have muscles of mastication and some ear muscles as well. So let's suppose it is supplying the muscles of facial expression which are derived from pharyngeal arches. Of course, it's an afferent. It's an afferent. And of course, it's special one. It's not general, it's special. But whether it is visceral or whether it is somatic, interestingly, it's labeled as visceral. It is labeled as special visceral efferent. This might be confusing because it is supplying the voluntary muscle. It is not supplying involuntary muscle of the viscera. It is supplying voluntary muscle of, here in this case, your face, which are quite superficial. So it should be ideally, I think it should be special somatic efferent. But the reason they call it special visceral efferent is that they are derived from pharyngeal arches or branchial arches, and these branchial arches, they are phylogenetically, they are related with the gills of the fish, and gills are the part of respiratory system of the fish, especially in the small fish, in which there is not so much complicated and sophisticated mechanisms, and sophisticated muscular system does not exist in some small fishes and phylogenetically earlier type of fishes. In those fishes especially, the pharyngeal arches derived muscles, they are only related with gills, and gills are the part of respiratory system, gills are the part of viscera. So by following that convention, even in human, even in other highly developed and highly complicated and complex organisms, we still continue to call them visceral. although they are even forming the muscles of facial expression, even forming the muscles of ear, but still because in fishes they are associated with gills, which are the part of viscera, which are the part of respiratory system, we still call them visceral. The second reason is that they are closely associated and they closely developed embryologically with our pharynx. So that is why pharynx is of course the part of viscera. So that is why embryologists call it special visceral afferent rather than special somatic afferent. But it is again the confusing terminology. So some great authors, some great embryologists, they just put a cross here and they say you don't need to stress so much about it, just call it brachiomotor fibers, brachiomotor fibers or brachiomotor neurons. Why all this fuss about special visceral and special somatic? By the way, interesting thing is that it is controversial. Some books, although their number is very low, only one or two book I have read, I think, they write it as special somatic efferent. Although it's very rare, some old books they do this, but all this fuss is resolved by some scientists, some authors. They simply call it brachiomotor. Motor mean they are of course motor, they are efferent, and brachio mean they are related with branchial arches, so they call it brachiomotor fibers. So by the way, what I want to tell you is that these are the brachiomotor nuclei, or these are the special visceral efferent nuclei. So general somatic efferent, special visceral efferent, and here is the general visceral efferent. All these are visceral. And by the way, why it is lying here, why it is not here? Well, to be honest, I don't know precisely that why it is here, but a speculative reason is that because there is lack of space here, so it is pushed here, it migrates here. Actually it develops from the basal plate, so then after the development it migrates ventrally, or the other way to see it, that the sum of these cells of basal plate, they move from here, they migrate from here ventrally, and here they will form the nuclei consisting of group of neurons that are forming the special visceral efferent types of nerve fibers. So these three nerve fibers are there. Now here we have the general visceral afferent as well. So we also need special visceral afferent as well. So where the special one will go? Of course, here too there is lack of space, so special visceral afferent will fix somewhat here. It will form actually a subnucleus here. So here it will form a subnucleus like this. So here you can see this main nucleus is divided into two parts. One of the part is general visceral efferent, general visceral afferent, and the other part is special visceral afferent. This nucleus, by the way, is called nucleus of tractus solitarius, which is involved in taste sensation as well as the visceral sensation, general visceral afferent sensations. We will learn these things in later lectures as well. And this is general somatic afferent. Then there is one more nucleus which is needed, and that type of nucleus is, you see, this is general somatic afferent. So we also need special somatic afferent, and that special somatic afferent, it brings the sensation from ear, the hearing and balance sensation from the ear. That also develops, and that develops very laterally. So it develops very laterally. Somehow it finds the space. Fortunately or unfortunately, it does not try to fit within these, or it does not try to migrate so much. So we will have to learn this, that it somehow fits here at very lateral angle, and that it consists of cochlear and vestibular nuclear complex that we will see in the upcoming lectures as well. And of course, if it was metencephalon, then there would be that rhombic lip as well, because there is no rhombic lip, so it must be the myelencephalon. So all these group of nuclei They form a column in the brainstem that we will also again see later, and beside these we have certain other important structures as well, unique structures related with medulla and pons and midbrain that again we will see in the upcoming lectures. But this is the basic idea. These are the type of neuron that you must know, and it's very logical. If you try to learn the neurons of spinal cord and then you develop the concept, then it will become very easy for you to understand. So let's just label it again for me and for you as well. General somatic efferent, and this is general visceral efferent, right? And this interiorly displaced is special visceral efferent or brachiomotor. Then we have the special somatic efferent, not somatic, it's special visceral efferent, and general visceral efferent. Then we have the, what is this, general somatic efferent, and what is this, special somatic efferent. So 1 2 3 4 5 6 7 types of neurons are there. Again, if you try to memorize all this, I am pretty sure you will forget within seconds. But if you take it up very logically and conceptually, then I promise you will remember it forever for the years to come. So learn it once for all. Do not try to cram up and do not try to stress your mind. That's a mental torture. Please don't go through mental torture. Learn it logically and conceptually. And of course, there is no fun in telling that all these nuclei, they are bilateral. So similar arrangement here as well. I have just labeled on one side, but there is exact similar arrangement on the other side as well. Now finally, one last thing that I would like to discuss, and then we will wrap up this lecture. Now here, this is the roof of, this is the roof of fourth ventricle, which consists of ependymal layer as well as this thin layer of pia mater. And this thin layer of pia mater, it becomes quite vascular. It becomes highly vascular, and lots of blood vessels invest this pia mater, so it becomes vascular pia mater. Now this roof, it consists of ependyma. Along with the ependyma, we have highly vascular pia mater. So this, both of these two layers, which are forming the roof of fourth ventricle, as well as of course later we will see other ventricles have also similar roofs. So these roofs are now termed as Tela choroidea. So Tela choroidea. Then what is so special about Tela choroidea is that this vascular pia mater, it will proliferate and it will invaginate inward, and it will form branching finger-like structures. And these branching finger-like structures, they will be again invaded by the blood vessels, and it will become rich in blood vessel. And this structure, which consists of tuft or finger-like projections, and these finger-like projections, they consist of pia mater, ependymal epithelium, as well as the blood vessels. They are termed as, these structures, they are termed as choroidal plexus. This one is one plexus, this one is other. Choroidal plexus. Now this structure, this roof is Tela choroidea. This is Tela choroidea or Tela choroidea. Tela choroidea is actually acting as a support, or it is actually acting as a scaffold for the choroidal plexus. Choroidal plexus are very important structures because they form the cerebrospinal fluid. Within this, they secrete the cerebrospinal fluid within this. Within these ventricles. Of course, beside the fourth ventricle, these choroidal plexus are present related with other ventricles as well. So Tela choroidea, choroidal plexus, this was all about this. So that was all about today's lecture. In the next lecture, we will see exactly that what are the structures that form the medulla oblongata and how do they develop. And then in the next lecture from that we will see the development of pons, then we will see development of midbrain and development of medulla, and ultimately we will learn all of the central nervous system embryologically. Thank you so much for watching this video. I hope you learned something from this. Have a great day. Goodbye.